everyone. Welcome to this week's podcast broadcast of This Week in Science. We are so great to be seeing you again this evening. No Blair tonight, but we've got a Justin. But apparently he's dying. I don't know. Yeah.
I've got a sniffly thing going on. I don't know what it is.
Sniffly thing.
I'm going to be muted for most of the show just so you don't hear me constantly inhaling snot.
Oh, my goodness. Oh, my goodness. All right, well, let us all begin this program. I love to tell you all that the beginning of the program right now, where we are all at, is the part where I tell you that there may be mistakes and mess ups and things that need troubleshooting. And when that occurs, you're going to watch it happen live. And we'll say, hey, Rachel, you got to edit this, or, whatever needs to happen. But this part of the show gets edited out for our edited program, which is a podcast that you can subscribe to. So make sure you look for This Week in Science, all places that podcasts are found. And that's the edited version. For the rest of y'all who are here for the video, let's do the science, warts and all. You ready?
Let's go.
Let's go. Let's go. What do I do right now? Oh, I click the things like this and I make it go like that. And I say, starting the show in 3, 2, this. Wait, what do I say? Yeah, this is TWIS. I sometimes get confused about the world I live in. It's been a long day. Okay, starting again in 3, 2. This is TWIS. This Week in Science, episode number 1006, recorded on March 12th. March 12th, 2005. Knowledge dies in silence. Why are you laughing?
2005? What the? Well, you're broken.
I am
Kiki,
so
can we get Kiki fixed? Kiki's broken again.
Somebody did a time change this weekend, and I have been a disaster ever since. I swear. When it comes to time, I have gotten every time zone confused. I've been sending emails all week trying to organize the conference, the science communication conference that I organize every year. Trying to get the virtual conference going, sending emails, confirming people's times that they're going to be scheduled and giving them the wrong time. No. I am a mess. Oh my God. I am in Rocky Horror right now. That's where I live. We're doing the time warp.
That's a good one. Yeah.
Yeah. Let's do it's a time warp, everybody. We're all broken this week. Exactly. Thanks for your patience, everyone. Starting again. I can actually do this. I remember what I'm supposed to do now. My brain will restart, rebooting in 3, 2. This is TWIS. This Week in Science, episode number 1006, recorded on Wednesday, March 12th, 2025. Knowledge dies in silence. Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your heads with new cells, plastic plants, and life in space. But first,
Disclaimer. This disclaimer would normally be here in this segment, but it's been replaced. I'd normally say something about the importance of science and society, yada yada, but has been replaced because as of the airing of this broadcast, the U.S. government has essentially incarcerated a student for practicing free speech on a university campus. We're talking about Mahmoud Khalil, who is actively involved in protesting the war on Gaza at Columbia University last year. You can agree with it, you can disagree with it, I don't really care, it doesn't really matter. His green card, which was in good standing, was revoked, and Khalil was sent to a detention center in Jena, Louisiana, presumably for being in the country illegally, though it was seemingly only revoked in order to arrest him. The reason he was sent to a detention center in Louisiana, not New York, where he was arrested, likely as a form of judge shopping, courts being more conservative there. But it also takes Cleo far from friends and family and lawyers, making his defense more difficult. As of now, it's been there for like a week and no phone calls. America! Students, citizens, visitors from abroad, be warned. This is not law enforcement. This is government opinion enforced by force. This is exactly what tyranny looks like. It does not happen in a free country. With that, I now bring us back to This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki.
And a good science to you too, Justin, and everyone out there. Welcome to this episode of This Week in Science. We're here to talk about science from the week and, maybe, discuss real truths about the world we live in, human psychology, also, no, we're not going there. But yeah, the world we live in, what a place these days. Apparently, there is some work out. There was a book written with a major study related to looking at the number of democracies in our world and those that had protests taking place to protect their democracies. Out of 394 of these protests that had occurred, it was discovered that when protests are able to be sustained and are able to crowd rise, to be able to build enough momentum to bring at least 3.5% of the population of that country into the protest, into the streets. When you have sustained building and up to 3.5% of your population protesting what's happening, those protests are successful in fighting off attempts at government takeovers by authoritarian regimes. Yeah. Anyway, that was there. There's an article in Big Think this week. If people are actually, no, not this week. The article's actually back from like 2019. But here in the United States, if we were to do that, we would need at least 11.5 million people across the country.
Across the country, though, as long as they don't have to be in the same place.
Nope. Not in the same place. Feasible. No. But 3.5%. It's a very interesting percentage, but that seems to be kind of the number of people where the people who might be in the military putting protests down violently, that they don't want to fight against people they know. They're more likely to run into teachers, parents, family members, other individuals from their community, and that makes it a little bit more confusing. But anyway, on that note, let's talk about science. That was a science study, social science, but we have a lot of very interesting news here in the show for you tonight. I've got space. I'm going to talk about space and people in space and stuff in space and whatever's happening in space, lots of spacey things. I've got some plant plastic problems and researchers trying to embiggen cells. Additionally, Alzheimer's and a way to stop speaking. What do you have, Justin?
That's a great question. I've got, is democracy safe? It's a different study on democracy countries. It's going to be a little unintuitive. Question, one of the question headlines. Do East Asians originate from a different human lineage than the rest of us? Do they?
Well, that's an interesting question. The braided stream, right? How does it all work? Yeah.
Or an unbraided stream.
Unbraided.
Completely separate origin story.
An unraveling stream.
An out of Africa story.
Yeah. All right.
for East
was
Asians.
talking about it.
And another way to look for life in, what would we call it? Space. Out there
Ooh,
somewhere.
you got some space stuff too? Let's look for life out there. Let's do it. I hope everyone is ready for the science. Oh, but first, I just want to remind you that if you are interested in all things TWIS, head over to twis.org. That's where we've got show notes and we've got links to our podcast files, our audio files. We also have links to places like YouTube and Spotify. And I don't think we have a link to Twitch. I'm not sure. But these are places where we put our live streams and our podcasts, and you can subscribe. We are live streaming weekly, Wednesdays, 8 p.m. Pacific time. Hey, Justin, I just realized you're here and it's not a time change and you're not early and you're not late. This is awesome.
Well, it kind of
It was a time change. It was a time change, but you weren't in a completely different country that does it on different weeks.
No, but I did lose an hour leaving my car today because my car still didn't know what time it was. From my car to here, I lost an hour.
It's like, oh no, it's 4 o'clock. Okay, I'll take it. Yeah, that's my microwave for the next 6 months. All right, everyone. Time for the science. Ready? Let's talk about space. Let's jump in off the docket with another space mission landing on the moon and falling over. Oh, poor intuitive machines. They have tried, this now is their second attempt to land a lander on the moon. And it is the second time that they have managed to kind of land a lander and then have it fall over and not work anymore.
Are they doing some ridiculous tripod thing? 4 legs. Give it 4 legs, maybe 5. Give up on the tripod if that's what's happening.
It's a tripod. So
is it really? Is it? Because I have never seen this
lander.
Thing.
Yeah.
I was just guessing. Because I've never understood a 3-legged stool. I'm like, it's gonna fall over. You're gonna lean on it the wrong way, and it's gonna disappear out from under you.
I have a 3-legged coffee table, and it is beautiful, and I love it. But I need to add more legs to it because if you lean on it just the wrong way, suddenly your water glasses are spilled everywhere.
Yeah, forget it. Put like 6 legs on that thing. Keep it on the ground.
Yeah. Well, anyway, it was they started. Things were great until they started to land on the moon. This Athena lander, they ran into trouble as they were going through the landing processes. It did land and then apparently toppled over, and they have declared it dead, unusable. No, not so good. But the good news from it is that they were actually able to receive some data before the mission was called to its end. They were able to capture images. And this is in a crater on the moon's south pole. Apparently, though, the lander it landed on its side. It's about 820 feet from the landing site it was supposed to land on. Not terrible. But the data that they were able to collect after the touchdown at approximately 11:30 a.m. March 6th, then they were able to confirm that it was sideways, and it couldn't do the drilling operations that it wanted to do, and its batteries were going to deplete because they didn't have any solar panels where they should be. And this is the first lander that is closer to the lunar South Pole than any previous lander, which is really great. They got images of the landing site. NASA has confirmed that they were able to actually see everything that was around in the landing site. All the stuff on the lander worked. So NASA's Polar Resources Ice Mining Experiment 1, which has a drill that's called the Trident, the regolith and ice drill for exploring new terrain, they were able to show that it could work, although it wasn't actually drilling into stuff. They also have the M-SOLO mass spectrometer observing lunar operations as part of the instruments. And it did detect some elements like gases maybe that were emitted from the lander's propulsion system. So even though nothing was able to really truly work and do what was experimentally intended in the South Pole of the moon, it made it there. the thing didn't bust up into lots of pieces. They were able to show proof of concept for all the technology, and that these technologies will probably be useful in future missions. Intuitive Machines has 2 more launches funded and approved by NASA at this point in time. So 2026, they're going to be trying again. And then there is a 4th launch, maybe in years after that. We also know that
I feel like this is just like battle bots. Okay.
Yeah,
you know
well,
what
this is
I'm saying?
right after the Firefly landing that was very successful. So we do have a commercial company that landed successfully on the moon. This is really cool. So yeah, it is kind of battle bots.
If you've ever seen the battle bots where people design these little robots and go fight each other, the one thing that's successful is having a mechanism that can prevent you from just getting flipped. Because there's one, that's how in the old days, that's how they start. They would just flip you. It was like a little spatula. You could have the buzzsaws and the drills and the things in the BattleBot. But the one with the spatula that just flips things would win. Mm-hmm. And then everybody designed it so theirs could work, upside down, right side up, didn't matter, or a way of unflipping themselves.
Yeah. Like some kind of a launch lever
that's
to
just
flip yourself
basic.
Over. Yeah.
That's just basic robot knowledge, is how to not end up turtled.
Right? It's like, okay, you have nobody to help you. Nobody will go pick you up. So what are you going to do? I'm trying
to be like, I love that they found like, oh, we got some pictures from the parking lot. That's wonderful. That's great. I love the effort to put a spin on it. But come on. Come on. It's like somebody in the chat room, perhaps close saying, yes, this seems so foreseeable. Yes.
Oh my gosh. All right. Well, moving on from the turtled Athena, we do have some data from that, which is great, but they're gonna have to try again. We do know there's another launch coming at long last this week. There is a launch that is occurring this week, March 13th, for NASA's SpaceX Crew 10 to head to, it may have happened already. A liftoff was targeted for 7:48 p.m. Eastern Daylight Time, March 12th, from Launch Complex 39A, and it will probably be docking at 6 a.m. Thursday. No, am I confused about days again? Yeah, no, I swear to God, what is a week? What are days? I have lost all of that. 6 a.m. Thursday morning, Eastern time, so 3 a.m. for those of us in the Pacific Northwest. Tomorrow morning, we will hopefully have NASA SpaceX Crew 10 at the International Space Station. And what are they going to be doing? What are they doing,
nothing.
Justin?
Nothing. They're not going to do anything because they didn't launch.
Did they not launch?
No, I googled it because I was like, oh yeah, I think that was today, and it says it was not launched due to hydraulics issues.
All right. Well,
the technology that they have on like old school tractors.
I was putting this story together. Yeah, I was putting this story together just prior to the show, so. We should actually check and make sure that this is happening. But the big thing, if this launches, if it's great, this week we will see the Boeing astronauts returned to Earth in the SpaceX craft.
The NASA astronauts, they left there by the Boeing test craft, the Boeing test craft pilots who got stuck
Boeing
there.
Boeing test pilots. Yes.
Because somehow or another we thought the Defense Department could do space. I don't know. Or airplanes for that matter. Yeah.
Okay. No. Crew 10 launch. I'm Googling it right now. Fly date, March 12th, 2025. Yep, you said it. They could try again as early as tomorrow. So maybe tomorrow morning. We'll see how it goes. I know this is going to happen. This is the longest saga, and I think it's kind of appropriate that the launch didn't go off at the time and date that it was planned.
But that's fine. And if I'm the lump of flesh in the giant firecracker, and any of those red lights go on, that's like, it might be a problem. Hold
launch.
off.
Scrub it.
Don't hit the button.
Exactly.
We'll just go triple check, make sure everything's plugged in and nothing's leaking, and try again later. I'm 100% for delays of this sort. I think they're that means somebody's doing their job, right?
These days, I'm all about delays, if that's what's necessary. One thing that did go off without a hitch this week, NASA's newest space observatory, SPHERE-X, is headed out into orbit around our planet, but it'll be looking at the sun. The SPHERE-X is going to be looking at our universe, our sun, and looking outward into hundreds of millions of galaxies to look at the ingredients of life that are out there. So we've got the SPHERE-X spectrophotometer for the history of the universe, epoch of reionization, and ICES Explorer. It just rolls off the tongue, but it successfully lifted off 8:10 p.m. on March 11th, yesterday, from Vandenberg Space Force Base in California. There are 4 small satellites as well that are called PUNCH, polarimeter to unify the corona and heliosphere mission. And that's going to be looking at the sun and its outer atmosphere and to figure out how the sun's outer atmosphere ends up turning into the solar wind, which impacts our planet and the rest of the planets in our solar system. This is very exciting that they're doing this. The mission is going to go through a period of time of checks and balances before it actually begins to collect data. But once it is working on collecting data, it'll be really wonderful to be able to see what it's able to determine about our own galaxy and the ingredients that are around all the planets in the asteroid belt, all over the place in our own galaxy, and even outward and beyond, to look at places that the James Webb Space Telescope has observed, or the Hubble, or any number of our other space telescopes. It just adds another tool to that complement of tools that allow us to be able to answer questions about what our universe is made up of and where is it. It's so clumpy. Our universe is so clumpy. We'll find out all sorts of things. You wanted to talk about life and finding life in space, though, yeah? Were you going to talk about SPHERE-X?
No. This is
something else. Fantastic.
Science. They've come up with this possible new way of detecting life on faraway planets, on worlds that look nothing like Earth. So we've talked about this before quite a bit, that we're very Earth-centric. We are looking for oxygen-rich planets, or planets that have water, rocky planets with water. We have this Goldilocks zone that we imagine the right planets to be. Well, atmosphere would help, right? So this is a new astrophysical journal Letters paper. Researchers from the University of California, Riverside, are describing gases which could be detected in atmospheres of exoplanets, planets outside our solar system, using the James Webb. They're looking at methyl halides. Methyl halides. No, that's not even close.
Halides. Methyl halides?
Halides. Gases that comprise a methyl group which bear carbon and 3 hydrogen atoms. Okay. So these are primarily produced on Earth by bacteria, marine algae, fungi, and some plants. So
I think the important thing to add, though, the halide part is those are halogens. Those are gas like chlorine, bromine. Halides are a very specific group of elements as well. So the methyl group is very specific, but then the halides are specific elements with specific properties. So, okay, sorry to interrupt.
Eddie Schweiderman, astrobiologist, paper's co-author, says, unlike an Earth-like planet where atmospheric noise and telescope limitations make it difficult to detect biosignatures, Haitian planets offer a much clearer signal. Oxygen is currently difficult or impossible to detect on an Earth-like planet. However, methyl halides on Haitian worlds offer a unique opportunity for detection with existing technology, adds Michaela Lung, who's the first author on the story. So they're working on a way to finding life on other planets beyond our sort of Earth-centric view of things. And I think that just makes sense. Chances of another oxygen-swilling planet full of mammalian vertebrates, just, yeah. Oh, you're muted, Kiki. You're saying something.
Yeah,
very
not just
interesting.
mammalian. Yeah, not just mammalian vertebrates, but there are lots of organisms on the planet that are oxygen swilling and not just oxygen producing.
And only like sort of by accident, like the first rounds of life weren't.
Mm-hmm. Right.
Oxygen was actually poison to them. It was the first extinction was the rise of oxygen. So.
Right. So photoautotrophs? We have to thank the early fungi, bacteria, others that led to plants for actually putting oxygen in our atmosphere. But yeah. I wonder if though there's a point. So I guess it would be there's the point in the evolution of a planet if the circumstances are correct, that if the situation is correct, that a switch, basically it's like, oh, if the right things happen, then something can take advantage of the oxygen that's been created and not just those other byproducts.
And the thing is too, they're pointing out like this could be on multiple planets.
Yeah.
Like apparently this is not something that we're tuned into looking for right now. So while we've kind of like, oxygen's just too tough to see. Unless, it's an exact right orientation, I suppose, for us to view. Get a spectrum on it. This is not something that we've been specifically looking for, so they're throwing this out there as an option. And that would be like the sort of funny thing. Then it turns out there's we're finding dozens of planets that have this signature. Life is abundant in space now. We're looking at it wrong. Because we were too Earth-centric in our thinking.
Why would we be so Earth-centric? I mean, come on. What are we?
We have a sample of one.
Earthbound humans?
We have a sample of one, and I get it. That's why. And there's some things that we can reasonably expect. I mean, one of the indicators we have that there might be life on other planets is that we keep finding the fundamental building blocks are occurring on meteorite materials. So the building blocks for life should be somewhat the same out there. But where it goes from building blocks, without the instructions come later. The instructions, again, DNA. Yeah. Really, it's a list of ingredients more than a plan.
That's a much later, more complex information transfer protocol, right? DNA it's different. Yeah.
However, that backbone, that double helix, I would not be surprised to see a double, if not a triple helix in a exoplanetary life form, just because that design keeps things from breaking apart. Yeah, pulling apart or breaking apart, and it's just engineering wise,
it's more resilient and stable under
it's the stable
when it comes
system.
To evolution, not evolution, but to radiation and to all sorts of other assaults.
So I would expect that part could be very much the same. But oxygen breathing is such a, and even water in abundance, like on an ocean, not submerged water or moisture in soil. Those are such like way later down there. It's like, whoa, we're looking for life that has a strong Wi-Fi signal. Okay, you can, you're skipping a bunch of possibilities if that's all that you're starting with. Right?
Yeah. Look at all the things that it could be. Things ended up a particular way after millions of years of evolution, billions of years of evolution here. on Earth. Billions of years of evolution. Things started, they didn't work out. Try again. Start, and then finally, this one thing took off. Seemed to work really great. But it may have, maybe other planets, they're still going back and forth, up and down, things trying and going and using energy and creating systems, chemical systems. And then, maybe what time in their evolutionary path do we catch them in when we take a picture, when we look at the data right now, this snapshot? Yeah. Cool. More markers. Let's go find them. Let's go find all of them. Moving on from alien life, let's talk about plants. You like plants, right? What are plants good at, Justin?
Being eaten.
Yes, they are. What allows them
photosynthesis?
Yes.
Is that what you're
plants.
going after? Okay.
I was going after photosynthesis. But your point of being eaten is very, very important to this particular story. Researchers just published in the journal PNAS, a global estimate of multi-ecosystem photosynthesis losses under microplastic pollution. So we've been hearing about microplastics ending up in our bodies, in our brains, that microplastics are everywhere and inside everything. Well, they're also in the plants that we eat. However, when they get into the plants that we eat, what do the little microplastics, how do they interact with the systems of the plants, the metabolism? Research has shown that, particularly this study looked at how much photosynthesis and crop yield decreased with microplastic exposure. They found that first they used a machine learning model using current microplastic pollution levels, and they showed that this exposure reduces the chlorophyll content of plants, photoautotrophs, by 10 to 13%, 10.96 to 12.84%. They were then able to transfer that to actually see annual losses within real plants. This was not just a machine learning model, but they were able to then predict losses with different amounts of microplastics. And they are estimating annual global losses of 4 to 13.5% for main crops and 0.31 to 7.24% for global aquatic net productivity. So basically, the more microplastics that we are letting into water, the environment, they're being taken up by plants, and by those plants are then, their chemistry is getting messed up. And yeah. Okay. So what?
So when you say productivity,
primary productivity, yes.
So that is converting the sunlight into carbon into biomass, which is food and energy for the things that live in the water.
In the water or the ground. This is for crop yields on the ground and in the water, but there are different results depending on the ecosystem that they're in.
Right.
But yes.
And I guess I'm less concerned with on the land. Even though that's where I live and that's where people are going to have food problems.
Yep.
Because there's also a story here about eukaryotic phytoplankton declining due to ocean acidification.
Right.
So higher CO2, less primary production, less converting of that carbon from the carbon dioxide into, again, the organic matter for other aquatic species. I mean, when we're talking about other eukaryotic phytoplankton, which means
that's what I meant.
Less reproductive going on.
Yes.
Which means less of them. This study that I looked at showed about a 10% decline in these sort of subtropical areas where they were investigating, if they extrapolated it across. And what that basically means is there's two cycles. There's a carbon cycle that that carbon is absorbed by the ocean, and that's 11.
We call it a carbon sink. We like to think that it's like a sponge, right? The ocean's like, I will suck up all the carbon.
Yeah,
it can only do so much.
It does about 11 billion carbons, which is almost
11 billion carbons.
Tons of 11 billion tons of whatever it is.
11 billion carbons.
What is it? It's megatons. What is
That's funny. Yeah.
I gotta find the right wordiology here. But so it's 5 billion metric tons.
Metric tons. Okay.
Metric tons. Okay. Metric tons is what we're working with. So the ocean normally takes in about 11 million. But that cycle within the ocean is converting about 50 billion metric tons. And this is saying it could drop it by about 10%, so about 5 billion metric tons less in the carbon cycle, which means less of the things that are normally 10% less of the things that are normally fixing carbon in the oceans, which means it's like basically what it's saying.
This is a feedback
these two studies.
Yes, that there will be the fewer acidification because the ocean is getting more acidic. It cannot support the life. And then you add the plastic. And so you're reducing the amount of productivity, which is actually the sponging up of that carbon dioxide in the creation
disrupting the carbon cycle within the ocean.
Yeah, it means less
it's not doing the sinking.
It's not doing the sinking. So
as efficiently.
Yes, that 11 billion metric tons would be less. That's taken out of the atmosphere. The amount of oxygen that's returned to the atmosphere would be less. The oceans would acidify maybe more rapidly. Maybe, I don't know, maybe it would maybe this is an asymptotic kind of thing over time, but yeah. Not good, everybody. Let's soak up those microplastics before they get to our oceans. All right.
I guess it's not that the oceans wouldn't take up as much. It's that they wouldn't get sequestered once it's taken up, which means that the acidification and the pH changes, which means that the fish move and they die, and then the phytoplankton that they eat in the first place and the food chain dies. And then we're just living on a dead planet, and somebody far, far away is taking a signal from space and going, hey, look, we found a planet that's the only thing that seems to be living there is fungus. Mold seems to have crept over the entire surface of the world. A high carbon digesting mold has taken over that planet. Should we go there? Nah.
No.
We'll keep looking for life elsewhere. Plenty of those around.
Mold is life. I mean,
yeah, but I'm under my conjecture.
Yeah. So microplastics, we've wondered. Microplastics is one of those questions. It's like, what's it doing, right? Is it just like garbage where it's like, oh, it's a plastic detergent container that looks bad and it's just in a dump? Like, is it just that? Or is it that these microplastics are getting into the systems in a way that are impacting hormones and impacting metabolism and actually having impacts that people have been suggesting for a very long time? And unfortunately, the research has been very scarce. And it's only in the last decade or so that we have been really starting to see the impacts of these microplastics on biological systems. We're seeing how they affect reproduction. We're seeing how it's affecting the ability for, in this case, plants to photosynthesize, photoautotropes to make use of sunlight and take in carbon dioxide and turn it into biomass. And that is something that we all really should consider. It perhaps will influence the biomass in crops that are produced for food on the land surface, but like you were bringing up, Justin, it will definitely have a much broader impact in the oceans, where there are many more food. The food web is so much more impactful, and the oceans themselves are so important to life on the entire planet. Let's see. You don't have another story right now. You wanted to talk about that one. And so let's move on from plastic-powered plants to let's talk about some bigger cells. So what do you do? Have you ever played with a microscope, Justin?
Mm-hmm. Yeah, absolutely.
Yeah. And you know, how do you usually, like, when you want to make something in the microscope look bigger, like, what do you usually have to do?
Oh, you have to switch that three-legged viewing lens to get to the more magnification, or get one of those fancy high-resolution digital ones with an incredible amount of resolution. You just need to magnify and magnify and magnify until your poor, weak eyes can see it better.
Right. But one of the issues is that many times, even though you're magnifying, you kind of get stuck at a point that it trades off like the high resolution and depth of focus to be able to get actual images. And so a lot of labs, they can afford high-end microscopes. A lot of people, they try to, but they're more expensive. You have to really get there to be able to process all the information, to be able to develop the systems, to be able to actually use these really high resolution, good depth of field microscopy setups. Low-end microscopes, a lot of people can use them, but they're not as good. And
They're
so
limited.
they're limited, right? And so there's Assistant Professor Kevin Cox at Biology in Arts and Sciences at Washington University in St. Louis. He's an assistant member of the Donald Danforth Plant Science Center. He and his team have developed what they call expansion microscopy in plant protoplast systems. And this is the fun part. Instead of switching your lenses, your eyepieces, to be able to get a change in your resolution, your change in your microscope power to zoom in, it enlarges the biological tissues by putting them into hydrogels, so that this is the same stuff. It's hydrogels. It's stuff that's used in baby diapers to absorb water. The hydrogels are used in those little gel gun little beads that absorb water, and they were originally developed for the USDA. So what's super fun is that this particular methodology is expanding the cells themselves, taking the cells, and instead of using higher resolution magnification, it's making the cells themselves bigger. Basically saying, I can't see you. Grow up. Get bigger. And so they expand the cells using this hydrogel, and in doing so are able to allow people to be able to see the cells better. You're able to, with the exposed system, visualize chlorophyll, other aspects of the cells themselves, to be able to do a lot of really good science that would be not as easily accessible unless you had higher end microscopes. So this allows better visualization of plant systems for study, and you don't have to buy really expensive lenses and microscopes. You just put hydrogel in the system and can basically grow the cell itself. And the images that they have in this study are just absolutely beautiful. They're they've done some great work in coloring their images and being able to create images that make you really go, I want to do that too. And I hope that this is something that can be used in less well-funded labs and in in high schools and college laboratories, you know, around the country. I think it would be pretty cool. Yeah. When you can't see it, just make it bigger physically. That's what they've done.
Yeah, I've heard about this, doing this before in mammalian cells, I think.
Mm-hmm. Yeah, it's been used in animal research, but in plants, because of their rigid cellulose structured cell walls, they don't have uniform expansion. And so in this particular case, the innovation is that they used protoplasts, so plant cells with their walls removed. And so so basically probably digest the walls, the cell walls of these plant cells, and then use the exposed system to grow the cells without those cell walls, because then you can have the better view. Yeah. They figured out how to do it. Isn't that cool?
Very cool.
Yeah. This time it's I think what's really neat is being able to like modify techniques like that, where it's like, okay, we know it works in this system. How can we take it and make it applicable to these others? So new ways to do all of it. How can you take TWIS with you from one situation to another? How can what we do be applicable to what you do? I hope that you enjoy This Week in Science and that you share it with your friends and that they enjoy the same parts of it or even the different parts of it, and that you can all have very wonderful, compassionate, respectful conversations, very healthy dialogues about the science that we discuss in the show. On This Week in Science, we have not taken advertising funding for the podcast. You might see, as you're watching the show, advertisements on YouTube and other places just to do minor support. But I'm going to tell you, it is minor. What we really, really depend on is support from viewers and listeners like you. So if you enjoy the show, not just share it with other people. I mean, that's great. Grow our audience. If you can find a bunch of really awesome people to share TWIS with, that is a huge help. But if you can help support the show through becoming a member over on our Patreon community, that would be absolutely wonderful. $10 and more a month, and we will thank you by name at the end of the show. There's t-shirts, stickers, other fun things involved there too. And additionally, you head over to twis.org, where that Patreon link is. Go to our Zazzle link, and there you'll find merchandise that is specifically TWIS branded, but also a bunch of art from Blair and her TWIS Animal Corner calendars from the past several years. You'll see all sorts of wonderful art on pillows and blankets and shower curtains. Find the things that tickle your fancy and help support TWIS in the process. We really can't do this without you. Thank you so much for your support. All right, everybody. I would normally be saying it's time for Blair's Animal Corner, but we don't have a intro theme song for this next section of the show, which is Justin's Rant. No? Yes, no. No? Just science. Okay, here we go. What you got?
I got a coffee maker going right here. So hang on, give me.
I can't even hear it.
1 minute. Okay, well then never mind.
I can't even hear it. Go for it.
Okay, all right. So let's start off with the first one. Are East Asians from a completely separate lineage of human than all of the rest of us? And the answer is no. Of course not.
Of course
what a
not.
ridiculous question.
I was waiting.
Yeah. University of Tokyo researchers have investigated the origin and dispersal scenarios of Homo sapiens into East Eurasia. And the team examined how migration routes, genetic contributions from archaic humans, environmental adaptations helped shape modern populations, and have found an improbable flaw in one of the origin ideas.
Improbable?
Now, if you weren't... Yeah, so...
I can't hear your coffee now, by the way, but it's fine. Keep going.
All right. It's the slowest coffee maker I've ever seen. So the origin dispersal of humans, has been widely, although now much more narrowly debated, between two main competing models. Multi-regional evolution, which suggests that people were spread out everywhere and then evolved to become their modern selves, and its counter, the out-of-Africa model. Now, the out-of-Africa model is the one I think our listeners are probably most familiar with, suggests that Homo sapiens originated in Africa, then dispersed globally as pretty much completely formed current modern humans, and this remains the dominant scientific consensus over the past 30 years. It's older than that, but this is, it's basically once we got the DNA from people around the world.
More recently, that's been changing, yeah.
It's like basically 30 years ago when we did start doing global genomes that we started to realize hey, all of the people are one people. They all seem to be related to each other rather closely, with all of it sort of pointing in the direction of Africa. Okay. However, that is not the main belief everywhere.
That coffee sounds wonderful.
It's boiling over now. Okay.
Oh no.
So
the drama off camera is so tantalizing.
So multi-regional evolution proposes that modern humans evolved from archaic populations in multiple regions simultaneously. The major flaw I was mentioning before that the current study found with this multi-regional evolution is the utter improbability of the identical mutation leading to brain expansion occurring independently in multiple regions, populations around the world all at once.
Right. So not everybody could have had their brains expand. Like if everybody diversified and was going different directions.
We're all working with the same brain expansion.
Yep. Yes.
Mental capacity of humans is the same globally. And it's tied to the same genes globally. Which means it's from a global event. Or, it happened before everybody ended up everywhere else. Anyway.
The ancestor of your ancestor, my ancestor. And that's where it happened.
But this multi-regional evolution was the formative scientific belief considered in Western academia for forever. The British people evolved in Britain. People in China evolved in China, and, so on and so on. Okay.
This just makes me sad.
This idea, though, is still the most favored theory of evolution for East Asians amongst Chinese anthropologists and archaeologists. That's where the support for the idea that East Asians evolved directly from their local archaic humans. Peking man, Homo erectus, on into current East Asian Homo sapiens, rather than any connection to African Homo sapiens. This view is influenced in part because there's a lot of early archaic human fossils discovered in China. But it's also because it's a very nationalistically pleasing interpretation of human origins. It's a very China-centric view of it. Anyway, in the study, human dispersal into East Eurasia, ancient genome insights, and the need for research on physiological adaptations, published in the journal Physiological Anthropology. They went to the genes. Whole genome sequencing of both modern and ancient human populations. Mitochondrial DNA, nuclear DNA, autosomes, sex chromosomes, paleogenomic data from Neanderthals and Denisovans.
They looked at everything.
It all into this study. They put it all in. They also looked at some physical adaptations, the things like cold resistance, metabolic shifts, light sensitivity, all these things through their genomic variants. And, everything says, it looks like all non-African populations of humans share a common ancestor that looks like they left Africa approximately 60,000 years ago. Now, we know humans left Africa earlier than that, but the population that's outside of Africa now that survived is related to this 60,000-ish year ago exodus. There could be some elements of some early human out of Africa that's hiding in that data somewhere, but it's not the population driver for humans on the Earth.
Yeah.
They did also find some interesting sort of aspects of it looks like there was 2 migration paths into East Eurasia, a northern route that went north of the Himalayas and the southern route through this Indian subcontinent and Southeast Asia before reaching East Asia. And it finds that East Asians on the southern route are predominantly made up of what is now today Han Chinese, Japanese, and excluding the Jomon ancestry, and many of the Southeast Asian populations, with the northern reaches being more of later became the Native Americans. Took a completely different route.
Different route, yeah.
So, East Asians do not have a significant northern route of genetic contributions from that northern group, although Native Americans do have some of the southern route that got accumulated. They're mostly
Isn't that the interesting thing? I'm going to continue to be just so fascinated with that, because the idea that that humans left Asia and ended up taking a southern route to the Americas and ended up South America, moved throughout South America.
No, it doesn't mean that northern route, meaning they went north of the Himalayas, across the Bering, and then down into South America. But some of them, before they left,
some made a southern
they
route
met
as
up
well.
again with that southern route.
Yeah, we have, but there's the northern route through the Bering Strait into North America from the north down to the south. But there's other water routes that are suggested that.
But those water routes are just coastal.
Years ago. No, that's
those
not
water routes.
No, that's the thing that is suggested. That's the interesting part of it.
By who?
This was some work that was like last year, 2 years ago, suggesting that was looking at genomic data in South America and saying it didn't match up with some of the genomic data from North America. So people who are now living in South America and the indigenous populations of South America are not necessarily related to the North American indigenous populations, and that their genetic basis comes from maybe another path that people have suggested, kind of similar to the Polynesians crossing oceans, and that there's maybe some path. Yes, I know there's a problem. I know there's a problem with it, but this is so fascinating. This is the genetics. But this is not Polynesians either, so I'm getting it confused.
No, because the Polynesians were like much later. Like they weren't there.
No,
like there were canoes off the coast of California with Northern California Native Americans were running around with canoes before anybody set foot on a Polynesian island.
Yes.
Before there was a Hawaii.
Yes. I don't know.
Anyway, but yeah, there could have been. We could be talking about bands of this. There are certainly indications that people got in ahead of that glacial maximum, because there's just no way for the populations to have been there where they keep finding evidence of human population taking place, humans in the Americas, while the glacial maximum is in full tilt, meaning nobody's getting through that Bering Strait, and even those seas are pretty treacherous. And there's already people barefoot running around in the white sands of New Mexico, or possibly hunting
20,000-30,000 years ago. Yeah.
But
fascinating.
yeah, it's just more evidence that it's like the worst and the best thing is this, like, there's such a plethora of diverse archaic humans in China that it's like an amazing place for these discoveries. But it's also all being filtered. It's sort of like they took a scientist from the 1950s in America and sent them to modern day anywhere to go and analyze things. And that's kind of what is happening in China with their paleontology, or with their paleoarchaeology. It's filtered through this nationalistic view where they have to agree with what the government line is, and where the senior archaeologists are happy with their interpretation and they don't want to listen to anything else.
This is what we said. That's what we're going to say. Just listen to us. Yes. Yeah. I don't want to just listen.
But it's all there in the genes, and that's what's so fascinating.
And that's what we have to look into. And actually, I was just looking at South American ancestry, genetics and migration, and there are some really wonderful visualizations, infographics that are out there. And you're right, none of them are suggesting a water route, really, from Asia, Africa to South America.
It would be a water route along the coast.
The coast. That's what they're all.
From the northern. Yeah. Like there's been like a whole bunch of attempts also to, like, the Polynesian thing's been going on, I guess, for like 50 years, that they've been trying to call South Americans Polynesian. But there's also been an attempt to make, the Bering Native Americans the indigenous Japanese population that traveled there. And there's no the genomics just doesn't match, knocking it down. I mean, like, it's not, just isn't. But they did find also in this study, they found some contributions from archaics. The Denisovans look like they have contributed to some of the thermogenesis that helped for cold adaptation, some of the body fat distribution that you might see in the Inuit, as an example. They also found that the Denisovans, of course, contributed to the Tibetan people's high altitude adaptations. I think the Tibetans might have been northern route. Anyway, there was also found some Neanderthal genes that were associated with higher diabetes risk. They found, for some reason, these genes are particularly pronounced in Mexican and Latin American populations, which is also a very interesting thing. But yeah, it's an interesting mix of when you start to learn about the ancestry and how sort of the protective or diseases that we have may originate from some of our archaic ancestors, as well as just adaptations to there was a study, I don't think we covered it because it was a while ago, that was looking at skin pigment that found like the first light skin started to show up in Sweden, but very, very recently. Like even back in the Bronze Age, most people still were having pretty dark skin. So these adaptations that we see as all these differences between people and how all these things are like,
they're adaptations.
It's all the same people.
Yeah. What is there? There was one headline, and is it related to this, that East Asians with lactose tolerance, not intolerance, but tolerance, could have gotten that gene from Neanderthals.
Yeah.
And I think that's one of those very interesting little tidbits that's come out of this one.
What's this map you got here?
It's a map. I'm starting to do, a Google image search. South American ancestry, genetics, and migration. And so this is a map of visualizing a DNA study of the peopling of South America and where the first inhabitants would have come from. in South America. And this was probably about 2019, 2018 that the work was done, or that it was published. But 6 years ago or so, that we have ancestral types coming from the ancient Beringians heading out to the one group kept going north, went to Ontario. We don't know what happened to them there. No, the other group, like you said, took that coastal route and headed down went down through Central America, Central North America, down to Central America, down to Belize, down to the Central Andes, and down into South America. And it's just interesting to see where the ancestry kind of, where people went and how it all turned out. But there are other images that are just very cool. I love I'm enjoying these visualizations a lot. So I highly recommend this little search.
And so that coastal group
on
that ends
maps.
up in,
Yeah.
that ends up in, I don't know, where is it? Peru?
Mm-hmm.
Now that group would have touched the southern route East Asian population, because they have some shared genes there. Okay.
Mm-hmm.
Same with most of the North American, but that other South American group, like if that northern route starts crossing over before the East Asians make it all the way across India and Southeast Asia and then up again,
Yep,
that
You'll
could
have two
show
different
a
groups.
diversity. It should still very much link to that northern group, but this has always been the weird thing. Like that northern group is pinned to an almost 20,000 year group that's now extinct, and I can't remember what they're called, but the northern ones is what they're often referred. The sort of missing northern population.
The Beringians.
No, it's this is still
This unsampled
this is
population A.
this would have been still back in like Siberia, Mongolia, even. Like there's a northern population up there.
The ancient North Siberians.
Which was but they're yeah, they're not Inuit and they're not North American, but they're like related to them. The strong. Okay, so here's the weird thing, though. That population that's always been the ancestor is 20,000 years, and we now know that there's
it's older.
Americans, Americas have older indigenous population evidence, but that could still mean it was through that population that came over, but there could be population heading back the other way too once in a while. Like if you go this way, you go that way. Why would you only go east every day? Somebody was going west. And if they were there a long, long, long time ago, that could add to more confusion about this.
Yeah. I think it's fascinating. I think you're absolutely right on that, though. Yep. Different groups, different dates, different directions. And yeah. And it depends. Like, again, it's where are you taking the snapshot and what is your perspective when you do take that snapshot? And your perspective matters. So how does this relate to democracy?
Oh, what was the headline there?
Your headline?
Yeah. Is democracy safe?
Is it?
Well, this is a Norwegian University of Science and Technology study. And they looked at democracies. They wanted to. They
trust
set
the
out to find out.
Norwegians.
They set out to find out why democracies often have fewer murders per capita than other countries. Is it because democracies tend to be wealthier than other forms of government? Is it a sense of belonging together in this together sort of a thing? Is it that people get to vote more about what they want to see out of justice, police, social services, that sort of thing? Why is it that there's just less murder in democracies? So they took data from 171 different countries, analyzed it. This was like 30-year data for 171 countries to get as many of the murder statistics and that sort of thing. What they found was democracy increases the murder rate. Overall,
You're free to do
The number of murders increases by around 5% as a result of democratic conditions. This amounts to approximately
what does that mean,
0.5.
democratic conditions? Like, what is that specifically? So tell me about that. Yeah. Sorry.
If you get to vote for your leaders, then you have a 5% higher chance of having homicides than if you don't.
Why? This is so weird. Okay. What's going on, Justin?
The more democratic a country is, the greater the impact its form of government has to increase the murder rate. It says the worst cases are found in so-called egalitarian democracies, where there's a high level of equality among citizens. In egalitarian democracies, the murder rate increased by approximately 0.7 per 100,000 inhabitants. It's more than the 5%. The 5% was 0.5. So this is another.
Okay.
This is more.
Yeah.
This may seem counterintuitive, but the reason is, and then right there they almost lost me. Like, I don't know if you know what the reason is. But the reason is that democracies generally allow more freedom, including things like alcohol, drugs, and gangs. People go out more often, and there is less surveillance. Dictatorships or autocracies or authoritarians countries tend to have lower murder rates on average when when you factor everything else out, partly because there's a stricter social control, less permissive culture. This applies to example like less drugs, less alcohol, and things like
more
North
control
Korea
over
Arabia.
Over guns and more control over
more surveillance.
Yeah, you don't want people having guns because if they have guns, then they can revolt. That's here's the thing. This is why I thought this story was interesting, because you'd have to be like, democracy is dangerous. You got to own it, and that's just part of what we deal with, right? Yeah. But on the other hand, like, who are you trusting for data? Who are you trusting for? Like, what is your murder statistic within North Korea? Like, who is giving you that? Where are you getting good data on the Chinese stabbing rate? Like, stop it. So, like, on the one hand, I read it and I was like, wow, that's an eye opener. Gosh, of course, it makes sense. More freedom, more, liberty, taking liberty with your neighbors. Like, maybe that's it. But on the other hand, like, wait, how are you even getting information on murder rates from authoritarian regime? I remember what was the not the president, was it the president, or what do they call it in Iran? But he was at some UN conference. No, not the Ayatollah. That's the religious side of it. The government side is it Prime Minister of Iran or President of Iran? I can't remember how they do it. Doesn't matter. Ahmadinejad, whatever his name was. Yeah. Was basically like, yeah, we don't have gay people in our country. It's not a problem. Stop it. Nope.
Stop it.
Not true.
Stop it.
Yep.
Stop it.
Nope.
Yeah. So we have no murders. There's nobody kill anybody in this country. No, it's safe country. Everybody like each other. Stop it. I don't believe the data. That's part of the problem in trying to compare anything to a world of authoritarianism, is that you lose data credibility immediately. And that's going to be a problem. Hello, America. That's going to be a problem at home very soon.
Yeah.
If we don't do something. Because as soon as you get rid of government that can give information that contradicts what politicians are saying, because we've had watchdog groups, we have independent analysis of economy and environment and everything else. And we're losing it because it's being dismantled. And then the information is going to come from a political agenda. And that's when we lose any credibility. We've already had problems trying to figure out what the murder rate is in this country. Because it's not
And it's not
or
reported
death by
accurately.
gun, because we see somebody's deceased, but they don't always say how and that sort of thing.
And
We already
not
have
all, and we don't know there are the ones that are, there's no question about it. It's gone through the judicial system. It was assumed a murder. It's always an assumption until proven, right? And sometimes those are not proven correctly. Sometimes people are blamed for things that are accidents, not murders. So I don't, there's going to be squishiness in this data for sure.
Yeah, and it's like, data is only as good as you want to make it. Like, what is it? Finland is the happiest country on the planet right now. It's way so far up in the Arctic frozen north. Their suicide rate is much higher than sunny California, but they're happy.
Absolutely. Yeah, so is Colorado.
You have to balance all these things. But anyway, my point was, I loved this story because it was a shocker. Right? It's like, democracy is more violent than anywhere else. But then I also realized I have no idea if you can trust any of the information. There's no independent data collecting.
So if stuff is related to homicide as related to interpersonal violence, is there really less interpersonal violence in non-democratic regimes?
So great point. Like, is
what do you mean by
domestic violence isn't illegal?
Right.
Then what are you talking about? Then it's not a crime. Then it's not a statistic. No, okay, fine. Problem solved. Don't make it a crime.
I love, though, I'm looking at the paper right now, and you have the abstract, but then the very first sentence is a quote: "Crime is a social mirror," from a paper from 1999. It's just, and I think that's a very, just in itself, like what we're talking about, it is a social mirror, because what is crime? And that is based on what society deems a crime at a place and a time. That's really interesting. I immediately, while you were talking, I was trying to run to the end of the story to look at acknowledgments and funding and footnotes to try and get some idea of, is this paper, are they trying to push some kind of a spin? But it doesn't seem as though they are.
They might be.
This is the British Journal of Criminology. So.
Yeah, they might be. You never know.
Interesting.
It's the independent researcher who might have the bias. The Chinese anthropologists and archaeologists are ignoring 30 years of DNA data. The current administration in the United States is ignoring all climate data. In fact, they just removed funding for the military actions on Department of looking at global warming and climate change. Like, that's not something our government's for
defense.
Extremely important.
Climate.
Extremely important because we have also all sorts of bases that are now that have might be underwater. Like, we have a lot of where we're launching all those satellites from. Yeah, it turns out this might be underwater in a couple of years. That Cape Canaveral is not
Cape. Yeah, that particular NASA base, Cape Canaveral, several years back, I had a conversation with a climate researcher with NASA who was studying and predicting sea level rise and how it would impact the entire Johnson space, the Cape Canaveral, the space, and everything. Like, where's the water going to go? Where can they move things? What could they continue to use the facility for? How can they ameliorate? What could they do? So there's lots of work being done, but you suddenly get rid of the ability to predict and look at what's going to happen. You don't know where migration's coming from. You don't know, you can't predict when you actually do need to protect a border. Come on, everybody.
Do you know that the wildfires in Southern California, unprecedented, right? Hadn't happened like that before. Do you know a lot of those people?
The Santa Anas they've happened. There have been fires, yes, but all the conditions.
But nothing together, like on this scale.
Yeah.
So it's not a coincidence that a lot of insurance companies stopped selling fire insurance before the fire. After the fire, you would make sense. You'd be like, oh, yeah, there's been big fires there. You wouldn't want to sell it. No, they stopped it before because they can predict, because they're looking at global trends.
A week before. They stopped.
No,
they canceled things like within
the
week to the month before. No, there were several hundred people who lost their insurance in the week before.
Wow. Like they can tell when these things are happening because the conditions.
Yep.
And it's because they study. Like people are paying attention to these things. But we're not going to have. Anyway, I forgot how I was getting our attention.
This is your rant. There you go.
This is the rant.
Yeah.
This has been another rant by Justin Jackson. Now back to the show. What you got, Kiki?
Oh, I think I have a point. No, I have some fun science about our brains. You know, it doesn't matter what's going to be happening in the world. It doesn't matter where we came from. It only matters where we are going. And the rates of Alzheimer's disease and dementia are increasing. Woo! Isn't that awesome, everybody? It's probably microplastics. Kind of great. So researchers have been looking at the brain and trying to figure out, okay, what can we do to figure out how to treat Alzheimer's disease, as it is one of the growing numbers of degenerative brain diseases among individuals in the Western world. And as we've talked about before, people used to talk about various parts of the proteins that would get clumped up. And they had particular hypotheses that had driven drug development for decades. And then, oh my gosh, that's not something that works. Oh my gosh. Well, this week, there are a couple of studies out that are in a different direction and to me seem very promising. One is from the University of British Columbia's Faculty of Medicine, publishing in Nature Communications. And these researchers have kind of accidentally stumbled over a new type of cell in the brain. This cell is kind of egg-shaped. They call it ovoid, and so that's what they're calling it. It's like an ovoid neuron. And in mice, in the hippocampus in the brain, there is an excitatory kind of neuron that is called a pyramidal excitatory neuron. And this is one of those kinds of neurons, except instead of being like a pyramid, it's like an egg shape. It's ovoid. And they are adjacent to cells within the trisynaptic pathway of the hippocampus that is used for memory and for identifying things like your keys or people or a dog or an apple, for identification of things. And that type of identification and memory of objects starts to go as Alzheimer's starts to degenerate within the brain. And these new cell types that have just been discovered are really exciting because now that we have a different type of cell, it's not just, oh, it's a different shape. That's great. No, these cells actually have different molecular signals. Like they are using different receptors. They are receiving different signals. They are sending different signals. They are modifying that trisynaptic pathway and modifying the memories, making sure that certain memories are remembered or not remembered. So to be able to understand and actually have just identified a new type of cell within the hippocampus, that with this study, they did knockout mice where they were like, hey, we're going to get rid of those cells. We're going to stimulate those cells. We're going to, they did all sorts of manipulations to basically take those cells out of the picture and then also to turn them on and to get them into action when they weren't stimulated otherwise. And they could manipulate the memory within mice for different objects that the mice had identified. They could get the mice to remember to form stronger memories. They could change all sorts of aspects of it. So these cells, it was an accidental discovery. They were like, hey, what is going on here? These look different. Let's take a look at what's going on here. And they went down the rabbit hole And we're able to identify these this whole new population of cells, which is a completely new target to act upon, to look at, to figure out, okay, are these cells some of the cells that go bad, that when they get damaged the most, they're the ones that are influencing the degradation of our memories. And so now we can really truly start to ask those questions, which is kind of cool. It's kind of cool. And then additionally, on the Alzheimer's front, a separate group of researchers were looking at different proteins, and they're like, oh, let's look at the tau protein, which is in Alzheimer's. We've heard about tau tangles. These tau tangles are suggested to be very important for kind of mucking up the works of the metabolism of cells. And so when you have these tau tangles, the tau protein tangled in the wrong way, it starts to tangle other tau molecules, and eventually it just gets in the way. And then the garbage can't be taken out if you have mitochondrial issues, and all of a sudden a cell doesn't work. But which tau molecules are the ones that are important? Researchers over the years have identified a number of what they call isoforms, which are like kind of different copies. It's like, oh, you have a particular type of shoe that you like, but they have it in different colors, or this one particular type of, jeans, they have it in bell bottoms or straight legs or taper, that kind of thing. But it's a protein, it's the different isoform. And so these tau tangles, looking at researchers at the University of Cologne, they were looking at the taus in Alzheimer's disease and have identified a very specific variant out of a number of others using CRISPR-Cas9 and live cell imaging. They were using human-induced pluripotent stem cells, demonstrating that this one isoform, the 1N4R tau isoform, is responsible for the pathological effects on the cell. And they were able to look with the CRISPR-Cas9, basically go, we'll put this one in here. We're going to mess up this one. We'll put this one in here. And they tested it on a number of different, these induced pluripotent stem cells, and analyzed how each protein isoform affected a cell. And in this work, they were able to specifically identify that this one tau isoform is the one. It's the isoform that it's a problem child. It's the one that's really causing problems. And so potentially, because they're able to see this tau isoform as the issue, this could lead to better tau targeting therapies. Maybe there are other subspecies that are toxic that they don't know about yet, but it's just understanding what vulnerabilities there are in neurons related to tau, and then also the amyloid beta exposure. So you're exposed to amyloid beta, it twists the tau makes the tangles. But if you can keep tau from getting tangled, then you don't have and this is a word that they used, a tauopathy.
This sounds very much like the old thing that I think got debunked. It was just about.
But those were the amyloid beta sheets. It wasn't the tau. This is different. Okay. That was the amyloid beta, where a lot of the treatments were based on specifically like looking at these particular isoforms of the amyloid beta that in the research nobody could replicate. People were like, it's this one. Our amyloid beta sniffing dog has identified this particular molecule.
Might as well have been. But it was by
UCSF
a researcher who became and it was fraud. On and it was made up findings. Yeah, they didn't find it,
which is terrible. They didn't. And so this is one study, of course, but and we always need to keep that in mind when we're thinking about what a study shows or it doesn't show. But what the work can do is be part of the larger effort to be able to put all the methods out there, put the data out there, let people do their snooping and sniffing to see, did you fake your data? It got published, it got through peer review. Now let other people go, hey, it doesn't look like that worked. Right now, this study looks really good. And maybe this study is one that, maybe it's one that will lead to targeted tauopathy treatments. Or maybe it won't. But I think this in addition to new cell types, there's work going on, and I have faith that maybe one day I won't completely lose my memory.
If they can just come up with interventions, it doesn't have to be curative, just something that can intervene to just slow the process even, then it's completely worth it, and they should absolutely get funding to further research or have other places get funding to replicate. Because this has been going on for way too long. This is ridiculous. We have the technology.
I mean, like so much though. I mean, what is it? Oh, we're going to cure cancer. Well, sorry, cancer is that's a catch-all phrase. You're not.
But we have
it. But we have
cured cancers.
Yes.
Many times we've cured cancers.
Or been able to treat them better, right? We've improved our treatment options.
And cured and removed completely. We've done this. But it's not one thing. That one's many, many things. Alzheimer's seems like it's one thing. And for the amount of time, effort, and money that's been spent on it, I feel like we should have gotten better results.
But so this is the question. Things do get into catch-all groups, right? So we have Alzheimer's and dementia, right? They are not the same. Do they come from genetic predispositions? Maybe. So that means the starting point is different. Are they triggered, or are they instigated by environmental conditions or compounds, right? And so that is also potentially an interactive and combining starting point, right? You've got genetics and you've got environment. Those two things, right? And
there was a story.
But then we've started to dig into it, and it's like, but then we've started to dig into it, and we're like, oh, but wow, mitochondria. The mitochondria are going bad, and if you don't, or the lymph system is going bad. So how do we keep the garbage disposal system within the brain active, and what is causing that to break down, and what is the thing that actually starts the degradation process in the first place? And so, like something like schizophrenia, which again is kind of a catch-all, there are multiple types, and we see this in Alzheimer's, we see this in dementia, but in our cultural perspective, this is the thing, and this is the way it's going to end up. But from an actual scientific or medical perspective, when we're thinking about therapies, right, you got to be we have to go granular. But yeah, all the things. What did you want to talk about very quickly that you weren't going to bring?
This was a story I covered a while ago, and this was, I thought, very interesting because it fits in right what we were talking about when you mentioned environment. There's a genetic variant that is known to cause dominantly inherited Alzheimer's disease. People who get this, usually, I think it's by age 52-ish, by age 50, they pretty much all get Alzheimer's disease. They diagnose.
So it's like it's a younger onset.
This one is a genetic, and a younger. It's a very aggressive. This is a dominant one. There's one guy who was part of this long study, the Aaaand study, or what is it? Sorry, dominantly inherited Alzheimer's network, Aaaand study. Okay. Yep. Of the Aaaand mutation. Okay. So he didn't get it. They've been following this guy for 30 plus years. 50s come and go, he doesn't get it. 60s come and go, he doesn't get it. Age 71, he remains cognitively normal, despite his 13 maternal
Genetics.
aunts and uncles that all got it by age 50. And he's got the gene that they think is responsible. What was different about this guy?
Tell me he drank whiskey. No.
He was a Navy mechanic.
What?
He was a Navy mechanic.
That doesn't sound like a healthy, like, it's going to, like, that sounds like lots of grease and gases and, yeah, okay, keep going.
So, yeah, unlike his typical, there's typical tau progression that takes place. His was confined to the occipital lobe, and it never spread.
Huh.
So it just stayed there, unlike all of his relatives. But the thing is, he was a Navy mechanic. And they figured this out because he had a high preponderance of, what is this, heat repair proteins? Let me see if I can find out what it
heat
what
shock
it is
proteins.
Yes.
Yeah, heat shock proteins.
And they think that developed because at a young age, as a teenager and in his young 20s, part of his work as a mechanic caused him to go from hot to cold to hot to cold. He went to a very hot environment to do his work and would have to go through rapid cooling. It's like almost like an endurance type thing. He would have to keep doing as he did his work.
He's in a cold place, and then he's cold, and then he'd have to go into the hot engine room, and then leave, and it's cold.
Engine room, and then get dramatically cooled down his body temperature
because it was too hot. Okay, yeah,
because it was too hot. So they think that during that developmental stage of youth, he developed these heat shock protection proteins that have protected the rest of his brain. And now they're curious also, like, hey, in the area that wasn't protected, was it already there in his 20s? And now it's been it's been stabilized so it can't spread, but it's also because that's where it already was. That now deposition was already localized there.
Right. So did it in the occipital lobe spread it within it? So the occipital lobe is responsible for visual processing, spatial reasoning, visual memory. So did he have degradations related to visual context in his ability to function?
Apparently at age 71, he's nothing more unusual than like a 71-year-old would have, right? His cognitive skills are excellent. He hasn't lost a beat anywhere. But he's not blind or anything. Fellows are wearing spectacles.
Not blind, but not, maybe not able to recognize objects, right? But maybe that's the only aspect of memories that disappear. Maybe he's able to remember things, but then forget kind of what things looked like, or does he not remember how to get around and navigate?
No, he remembers everything. He doesn't have any problems. He doesn't show any of the signs of Alzheimer's.
Okay, so you meant, the reason I'm asking it, because you mentioned that he did have some buildup and degradation of the occipital lobe. So what I was wondering, like you mentioned, was it already there when he became this hot cold engine room mechanic?
That's what I was thinking.
And did the heat shock proteins completely stall the progression in the occipital lobe and the rest of the brain, right? So like you were saying, had it already gotten underway with the degradation, but not enough to cause like behavioral issues. And then he did this job, and was he protected?
So
now what are we going to do?
Cerebrospinal fluid and plasma biomarker analysis showed that amyloid B levels and amyloid B42/40 ratios were within the range observed in symptomatic dyad carriers.
So he had the amyloid beta.
Phosphorylated tau markers were intermediate between dyad carriers and non-carriers, suggesting potential modifications in the tau pathology. Mm-hmm. So environment, when you were saying environment,
yeah,
like that might almost have to be like the therapy Well, we're gonna
Sorry,
into the hot tub, out of the hot tub, into the hot tub, out of the hot tub.
Okay, so
We're
now
gonna have to
I
make
want
you do
to
that in your
know.
youth.
I want to
if
know.
you could.
Russians, Finnish, the cultures that do the cold plunge and banya every day. What are the?
It's not everybody. It's a few people they put on TV that do that. Like most people are like, still
Finnish,
not.
especially, like everyone's got a sauna. Everyone does sauna.
Saunas.
Which is hot.
Okay.
You don't have to do the cold, but everyone does sauna in Finland. It's like very regular. But the banya, going out, getting cold, going into the sauna, the banya, smacking yourself on the back with the fragrant stuff. But is the hot just enough? Or does it have to be hot, cold, hot, cold? Do you have to be a sauna and
whatever gets those heat shock proteins to hang out and get produced? The polar bear club. That's what we got to look at.
Yeah, so that's what I'm wondering. People who are like really who do this on a regular basis, do they have a different level of Alzheimer's precedent? Like what's their percentage of the disease in their populations? That's what I want to know.
I would definitely, that would not be protective for me. I'd get the Alzheimer's when I get old. I would so averse to the hot cold thing. The hot tub to the swimming pool even.
I am too.
Like a polar bear club.
Honestly, I will start doing it.
I don't know.
Do it every day.
I think it might have had to be formative environment. I don't know. Maybe because like the brain isn't done forming in your 20s, or young 20s, early 20s. I don't know.
But so right? Isn't that? But so that's the question, right? Is it the timing of the heat shock proteins? Number one, is it the heat shock proteins? Did that have anything to do with it? That is a big question. Yeah. I hope someone studies it. Second question, developmental timing. Is it before the brain is fully formed? We know that the frontal cortex, prefrontal cortex doesn't complete its formation until the early 20s. So it's very possible that was protected. And that's a really important area of the brain, especially with Alzheimer's for controlling your behavior, for more complex cognitive behavioral functions. I don't know. Now I want to know more. Oh, I really hope somebody does more study on this. This is so fascinating. Thanks for bringing that up. Yeah. And just in that moment, I stopped a sentence and I redirected myself, and then I started transitioning into this new story. And there's a lot of things that are involved with the onset of talking and also turning it off. Have you ever thought about switch? Yeah, there is. And that's what this study is about. And actually, you might think about people that in your lives who have trouble stopping their speech or the stuff they have to get out. And, there are, I'm going to say my son, for example, but, as you grow up, you learn how to stop what you're saying, wait for somebody else, listen, hopefully, to what they're saying and incorporate what they're saying into your response so that then you can respond more appropriately. And this is something that is done with training, but it's very important for your brain to be able to stop you from just verbally running people over. It's very, very important. So this particular study, that's what it was about. Researchers were looking into the premotor cortical network for stopping yourself from talking. Speech is supported by lots of motor networks. There's the thinking, processing of information, the understanding of words and concepts and putting them together in syntactical and grammatical fashion to get ideas expressed verbally. Those have to all be coordinated with your vocal cords, your jaw, your mouth, I'm speaking in body language, also using my hands right now. And so, there's this speech production process is incredibly important to social interactions. And it's also very important to be able to stop the talking so that you can actually have a conversation as opposed to be giving a monologue, I mean. So the researchers published in the journal human behavior, and found a previously unknown premotor cortical network to voluntarily inhibit speech production. So this isn't involuntary speech inhibition. This is voluntary. I heard somebody say something. I'm going to stop talking now. Or I'm talking, my phone rang. I'm going to stop talking, pick up the phone, and listen to somebody else. So the voluntary choice to stop speaking. In the study, researchers got epilepsy patients, 13 patients diagnosed with treatment-resistant epilepsy. They had electrodes implanted in their brains, and when they were going through surgical procedures, they were involved in this study. Patients were asked to complete a task where they stopped talking. Go, no go. And so they followed instructions on when to start speaking and saying various words and when to stop. Say something, don't say something. And they recorded the neural activity in the brain. And this found a very specific premotor frontal cortex circuit. Frontal cortex, this is what we were just talking about. It stops the speech. And the activity was found, according to the researchers, in largely separate cortical sites from regions encoding vocal tract articulatory movements. And this activity primarily occurred with abrupt stopping in the middle of an utterance rather than naturally completing a phrase. And so the researchers, when people, they not only were getting them to say things and not say things, start speech, stop speech, say something, don't say something. They also electrically stimulated areas of the brain to actually make utterances. So change the voluntary into involuntary stoppage of speech. So once they identified the network, they actually manipulated the network, which I think is very interesting. They say electrocortical stimulation at many premotor sites with inhibitory stop activity caused involuntary speech arrest, which contradicts previous clinical interpretations of this effect as evidence for critical centers of speech production. And the results suggest a previously unknown network. Yeah. So anyway, coming to you soon in the future, people probably trying to figure out how to keep you from talking. No, I don't know that. I'm just saying things anyway. Have we done it? Did we make it to the end? You're still alive. It's all good?
Yeah, yeah, I think we did.
I think we've done it. Thank you, everyone, for joining us for another episode of This Week in Science. We've been so happy to see you in the chat room, watching live, joining us for the conversation, enjoying the stories, commenting on them. I really enjoy watching everything that people have to say. Additionally, I want to say thank you to everyone who helps with the show. Fada, thank you so much for your assistance with social media and show notes. Gord, Aaaand, Laura, other people who keep the chat rooms good places to be. Thank you for being there and taking care of those places. Identity 4, thank you for recording the show. It's nice to see you back again this week. I hope you had a good week off last week. Rachel, thank you so much for the work you do editing the show. And of course, as always, it is time to thank our Patreon sponsors. Thank you to Robert W. Farley, Lauren Gifford, Dana Lewis, Eden Mundell, Alan Viola, Aaron Anathema, Arthur Keppel, Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Bob Coles, Kent Northcote, George Corris, Pierre Velazar, John Ratnaswamy, Chris Wozniak, Bagard Shefstad, Donovan Stiles, a.k.a. Don Stylo, Ali Coffin, Shubru, Sarah Forfar, Don Mundus, B.I.G. Stephen Albaran, Daryl Myshak, Andrew Swanson, Fredis 104, Skyluke, Paul Ronovich, Kevin Reardon, Noodles, Jack Bryan.
Hang on a second.
What?
Kevin Reardon, by the way, just so it didn't go unnoticed, your hot, cold joke killed me in the middle of the show. In the chat room, when we're talking about the Alzheimer's thing, which is a serious subject,
I'm going to scroll back.
I have a lot to look forward to because when I was younger, I had a girlfriend who went between hot and cold frequently. And I almost spit coffee out of my nose, I think. I was like, I wanted to just stop and start laughing in the middle of the show. I'm interrupting the show now to do it just because I can't let it go unseen. But very well done, sir.
Oh, dear. Where was I? Where was I? Oh, my goodness. Wait a second. Jack, Brian Carrington, David E. Youngblood, Sean Clarence Lamb, John McCree, Greg Riley, Mark Hessenblow, Steve Leeson, AKZ Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard, Brendan Minish, Johnny Gridley, Clemmy Day, Drew Putin, Lattimore, Flying Out, Christopher Dreyer, Artie, I'm Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rod Lewis, Paul, Philip Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, Lon Makes, E.O., Adam Mishkan, Kevin Parachan, Aaron Luthen, Bob Calder, Marjorie, Paul D. Disney, Patrick Beccararo, and Tony Steele. Thank you for all of your support on Patreon. We cannot do it without you. And if anyone here is interested in joining us over on Patreon, head over to twis.org, click that Patreon link, and I look forward to reading your name in this list next month. Also, I hope Patrick's doing all right. And Tony Steele, he is an amazing artist. You should go look for his art. On next week's show, Justin.
We will be back Wednesday, 8 p.m. Pacific time, broadcasting live from our Twitch, YouTube, and Facebook channels. Want to listen to us as a podcast? Well, you can. Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe too.
Oh, yeah. Subscribe. 2468. Who do we appreciate? You. Appreciating TWIS. You're going to keep
also,
going,
you know
right?
what I just found out? What? We are still on KDVS 90.3 FM in Davis. Where the show has been broadcast as a radio show for the last 25 years.
Did you actually hear us? Yay, KDVS.
Monday mornings.
That's great because I have been very bad about uploading our radio edits to the folder that was shared with me like years ago.
Uh-oh. Well, they're playing the same episode over and over again, maybe. I haven't actually listened to it. Who wakes up that morning, early in the morning? I don't. I do a podcast at night. Radio shows is a morning thing. Who knows?
Thank you, KDVS. You're our history.
KDVS.
Love it. Thank you.
And so for more information on anything you've heard here today, show notes, links to stories will be available on our website, www.twis.org. We love your feedback. If there's a topic you'd like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know on one of our social media accounts or send us an email. Just put TWIS, TWIS, somewhere in the subject line, or your email will be spam filtered into a satellite headed for the moon that will only have 3 legs and end up on its side, and we'll never get to know your lovely message. We look forward to discussing science with you again next week. And if you learned anything from the show, remember,
it's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. this week in science. Science, 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 science. I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and
Whatever. It's the after show, everyone. Justin has turned off his camera, so I assume he'll be back in a moment to say goodnight and all the things. It's 10 p.m. here in the Pacific Northwest in this new, is it daylight savings now? Is that what they call it in the summertime? Again, I cannot keep up with the time changes, and I am in rocky horror. Thank you, everyone, for joining us for another episode. I hope you're all doing well. Did anyone head out to Stand Up for Science events around the country this last Friday, March 7th? If you did, I'd love to hear from you and hear how it was. I have been deep in trying to organize this year's Science Talk Conference, which is happening in Raleigh, North Carolina in a couple of weeks. Oh my goodness. But I'm in charge of the virtual and our virtual conference, which starts next Monday. It's very exciting. We have so many amazing presenters and people talking about science communication. And I just can't wait for the whole thing. So if there are scientists and science communicators out there who are interested, you can be a part of the conference virtually throughout. Even though we're going to be in person in Raleigh, North Carolina, it's going to be live streamed. So it's hybrid. Yeah, I know. Thanks, Pepsi Cola. Yeah, take a month off to recover afterwards. Exactly. There's a lot. It's usually a sprint in this last month before the event occurs. And so I'm in it. There's a lot of details. And apparently this year I cannot deal with them. I am having issues with time. Who knew that would happen? But yeah, but there are events occurring around the country that are political that have impacted my conference, and so I think it's something that everybody should be aware of. We've had people say they can't come to the conference anymore because of changes in because of executive orders. We have people who do not feel safe talking about their research in panels at our conference anymore because of changes resulting from executive orders. The campus where our event center is located that university institution has changed their approach to language about diversity, equity, inclusion, accessibility. They have gotten rid of those words. They are changing the wording of things. And so we are, as an organization facing and as an event, trying to figure out how, with a limited budget, to be able to get people to the conference, even though they don't feel safe. How to create spaces within the conference for people to connect and talk about all the things that are happening. How does the science communication community move forward? And how do we address the challenges that are facing so many of us? People have lost jobs. We had a big list of volunteers and people that were going to become, and a lot of them have in the last couple of months lost their jobs, lost their funding. Yeah, so we need to have panels talking about all of that, right, Fada? Yeah, and we may add some panels. I don't know if the panels are the thing to do or if it's more creating rooms where there are facilitators so that people can have conversations. We're trying. And it's interesting because it's very similar to kind of when COVID happened and we had to flip from in-person to virtual for the first time ever and teach people how to be interactive virtually when there was not the infrastructure that there is now. So much has changed, yet it's applying things differently. But yeah, so there's a lot that we're trying to respond to and deal with at the moment. I just want people to be able to talk to each other and learn from each other and like make friends and maybe do really good things in the world. That's all I want. It's good people doing good things. Okay? That's all I want. But yeah. So anyway, the next few weeks are going to be a lot. I will be taking a week off in the first week of April because I will be in Raleigh doing the conference. And so I want to be all there and present for the conference that week. So hopefully Blair and Justin will be able to do the regular Wednesday show that week. I don't know. Maybe I'll find some times to like do live streams of conversations with people who want to talk during the week, because I think it would be fun to bring some of that to people here on this channel who want to hear about it. Because I don't know, the stuff that we do talking about science is so influenced. It is impacted by the money our government spends and the regulations that our government enacts.
Yeah. So we're funded by listeners.
By you. You listeners.
Look,
yeah, we can say what we want, which is
if you've
great.
been thinking about it, if you've been putting it off, if now's the time. Because we're going to start losing mainstream scientific communication channels. It's not going to affect us. But now more than ever, maybe if TWIS had a budget, we could start talking about a lot more of what's going on.
We could do more with it too. Mm-hmm.
Yeah. People are going to need a place to be able to communicate, because the frightening level. I mean, I've been on this planet for a while, as of probably a bunch of our listeners combined. We've span generations. This is different.
It is. It's different than. It's different, and coming from Denmark, right?
The attack on academia. The Department of Education being dismantled.
Cut in half. Just cut in half. 1,500
scientific
people.
Research being defunded. I mean, these are the investments that, regardless of what the political atmosphere is, even between countries, science has continued to march forward.
And it will continue to march
Education
forward.
has continued to be a priority.
And there will be new funding sources, and researchers will figure it out, and people will figure it out. But if the government censors information and databases, and
that means you can't use government as funding, and that's how our system is built. So so much of it. Yeah.
I don't know.
This is gonna be,
Don't make me take out my little tiny baby knife. It's very tiny.
Goodness. Oh my gosh, I think I had that when I was a kid.
It's
very similar. Oh, not that one, no. Oh my gosh.
It's so dangerous.
It's a chopper.
It's actually very sharp. Because Marshall sharpened it. Of course he did. But yeah, this is different than 9 years ago. This is different than when the climate stuff started happening decades ago. Right. There is a difference in what is happening now than what has happened
under George the W. There was censorship taking place within NASA, and there was pushes to get NASA to stop reporting on global warming because we had an oil cabal running government. Okay. Transparent in that we knew what their objective was and why, and the research continued. This is different. This is defunding all of science to stop a communication of fact and truth.
Yeah, I don't know if people. So another ripple outcome this last week that came about, that I learned about on Friday, I think, was that the National Association of Science Writers and the Council on the Advancement of Science Writing, they have an annual conference that usually partners with universities, because what they do is they bring science writers who write all your cool science articles in the New York Times, Popular Mechanics, websites. Yes, you're a science writer. I've been a member. I've been to the Science Writers Conference. for years. I just went to the Science Writers Conference in Raleigh in November. Got to meet with my colleagues. We were talking about all sorts of stuff. The 2005 Science Writers Conference has been put on hold because
2025.
What year is it? 2025.
2005 stuck in your head.
I don't care anymore. Years, time, doesn't matter.
2025. The
Everything
conference
is
is put
now.
on hold?
The Pittsburgh and Carnegie, that were the universities that committed to supporting venues and funding and supporting the conference, have pulled out.
Well, I wasn't going to go to it, but now I want to.
There is no conference now because they have no
because there
place.
isn't one. Now I want to go.
Right. No. There's no place. There's no funding. And so there are definitely big conversations underway right now across the board. People are picking up and trying to figure it out so that there will be a conference. But it's just amazing that because the researchers have had funding pulled, there have been grants pulled, there have been restrictions on a lot of things. These universities are now struggling to figure out their budgets and basically having to say, we can't support this thing anymore. Even though it's a great thing for their university because it'll bring science writers to the university to learn about the science that they're doing. So, but oh wait, no, we're not actually doing science anymore. It's not funded. So yeah, anyway, yeah, it's this last week hit really hard. It was like at the same time that Stand Up for Science was happening, this news came out, and I don't know. I'm just generally reeling. I'm looking forward to my conference. It'll be really good to get together with.
This is also why I started off the show.
This is where we got to find our 3.5%.
This is why I started off the show talking about Mahmoud Khalil, because this is also somebody who is legally in the country.
Yeah.
Who is a student who is just doing the American free speech thing on campus.
Yeah.
If they can just cavalierly revoke a legal protection such as a green card, I don't feel like there's much of a threshold between that and protecting the citizen status of free speech.
But even beyond that, it's not just the citizen status of free speech, it is the control of knowledge and knowledge production. And as much as, I mean, by dismantling and hobbling the funding infrastructure from the government for science, we are going to see people leave our country. They're going to go to Denmark. They're going to go do research and find positions in other countries where there's funding. If it's not happening here, they will find an environment. People will find an environment to do the work, and it's going to, like Lost Algorithm over in Twitch said, it's going to impact the research environment for over a decade. It's like there is a long-term impact that could be. In fact, yeah. Hey, Denmark's nice. It's really nice. Justin knows that very well. But
it's nice for the 2 weeks of summer, and then it gets cold and dark.
But suddenly the researchers are in Denmark. Intellectual property is at universities in Denmark. Technologies and medical treatments, all sorts of things that would be valuable here in the United States are now in other countries. And so maybe, suddenly that brain trust that leads to the technologies, the companies, the driving engine, we're going to end up on a 2-stroke if we're not careful. Like, it's not even going to be no injection in that engine, no sport function. We're going to end up like on a 2-stroke engine, barely getting rid of the weeds. So this is, China would get the researchers. Historically, we've gotten Chinese researchers. They've gone back home and then advanced the research in China, not necessarily staying here. A lot of people have stayed here, of course, to do the research and have jobs and do all sorts of things. But suddenly the migration flow is going to be reversed.
Well, I don't know. I find that less likely. What I think the scenario is.
What they're getting rid of the H-1B visas, or cutting down those numbers, which are student visas for people from other countries. They're employee visas for people from other countries.
Okay, I see what you're saying.
We have universities where they can't give students money to do research anymore.
Yeah.
And people are going to go where the money is.
Well, it's much more likely that when you're getting rid of the government funding of these things, it's very likely that a lot of this is designed for people to go into private research.
Yes.
That is going to.
But you don't get into private research
owned
until, but you don't get there until you've done a graduate degree. You don't get into one of those companies. You don't get into a George Church funded company, or, one of these big research biomedical, or, new AI, or even, the fusion company stuff. You don't get into the private companies until you have an advanced degree. And where are you going to get the advanced degree if nobody's giving money to pay for students? Because if you don't have the government grant, you don't have the money for your lab. You don't have the money for your lab, you're not paying students. There's no paying students, you can't have a graduate student. You don't have a graduate student, that graduate student is someplace else, probably another country.
Well, right, but that's the way the system's been. But what I'm saying is, with the likely long-term, if there is a long-term strategy,
I know, but this is
to convert all of that system into private, so that it's all of the intellectual property is owned.
But that's long term. Yeah, all owned by corporations.
Yeah,
where the universities even become private because they cannot do public funding anymore. So that's the next step. Yeah, you're right. But
on the other hand, the danger of taking away all government money from these universities is that then they're no longer beholden to anything the government thinks at the same time. Like, to a certain degree, like all of the diversity things that some universities added that they're now taking away, is because they still have concerns about their federal funding. Take too much of that away, and then, hey, you know what? We don't need you because you're not there.
Yeah. So the question Zombie Tom Hanks is asking is, do we want corporate only backed research? And the issue is no, because corporate research is based on profit. And so you do not get the investment in basic research, which is the investment on questions that are questions for the sake of questions. They're questions for the sake of following things to see if you can figure something out. And the government, by funding that basic research, is funding the foundation of knowledge generation, because it is that knowledge, that information, that allows people to make more informed questions, to form those questions and create those studies, and to get from that just inquiry-driven process into something that is more led by application. But you can't get rid of the foundation. You get rid of the foundation.
You're talking about the foundation because what you're talking about is like CRISPR-Cas9 came out of university research.
But it wasn't. It came out of research that was just random bacteria, and this was research. Like, this is stuff from global stuff.
The mRNA vaccines came out of university research.
But basic research first. It came. But I'm saying this is like people going, oh, I wonder what does this little gene that we found in this bacteria do? Yeah. What is this? And you don't get that in corporate research, because corporate research has to have a profitable function. And so you cannot ask questions.
What client does it serve?
What money is it going to make? What is the return on investment? Like, you get big behemoths that have like their quote unquote innovation labs. But like, honestly, the innovation that comes out of those innovation labs is questionable, because it's still based on profit. And you have to be an entrepreneur. You have to be acting as if you're gonna make a business out of this question that you don't even know if it could solve a problem.
Although technically
It's
that
just
is the Danish system. What you just described.
What do you mean?
From what I could tell, Denmark is made up of several thousand startups, each with a niche research project that they are trying to build a business on.
But this is the problem, this is like Silicon Valley, it's that startup culture. It is a solution in search of a problem, right? Yeah. And so many of them, maybe 99.9% are going to fail because
but that's also with every drug that's made.
But that is like following actual like you've already gotten to the point where you know there's a problem when you're making a drug. You've gotten to the point of like even the the phase 1, phase 2 clinical trials kind of stuff. You already know you've got a foundation of research that came from somewhere else that had to be funded by the government or like, some researcher who, was able to spin a phrase and convince a private philanthropy or funder to accept that this research on zebra finches was applicable to women's hormone replacement therapy. I mean, what? How?
The foundation of all tech and all medicine is government funded research at the basic level all the way up. That's how we did it. That's how we've done it.
The DOD, the government, NIH, NSF, these organizations fund basic questions. In the last few decades, NIH and NSF have become more application-based, and there's been a real pushback to it because it has changed the way that universities create information and create knowledge. And it's something that people think, and very rightly so, that it is negatively impacting the practice of science because it is creating generations of scientists who only do science for application purposes.
So the thing is that then you got to like, if it's anti-science, anti-education, with all this tariff stuff, it seems like it's anti-business, it's anti-trade, it's anti-American. What is it for? Anti-taxing, but also anti-spending, but also anti-investment and
absolutely
anti-democracy. It's anti-free speech. It's anti. What is it? What is it at all? What is it at all for?
If you take us down,
see what it's for.
Making up a song. If you can't see it.
Anti-logic, yes. Anti-logic. All right.
No.
I'm going to go blow my nose for the next 4 hours.
Go to bed, Sniffly. This was actually a very fun conversation here at the end of the show. I hope y'all enjoyed some of it, but it's a big deal what's happening right now, and I really hope that more of you look into stuff that's happening and what's going on. Don't just, of course, as we always say, don't just take us and what we're saying at face value. Go do your own, go ask your own questions and see what you learn and what you think after that point. But seriously, okay, my new favorite song. I have a new favorite song, and I found it randomly. And yes, I do still do Spotify, as people think Spotify is evil. But this song is by, and you'll love this, Justin. I'm looking because I shared it with people, and it is called, hold on, you can't leave. Is That All There Is? It's a song called Is That All There Is? And it's by, is it going to load here? Come on back. I'm going to see if I can get it to open. And then this whole, Peggy Lee is the artist. And I ask you, I'm not going to play it, but I ask you to listen to the whole song. This is my new favorite song, and I listen to it now every day. And I asked myself, is that all there is? Is that all there is? My friends, let's keep on dancing. Is that all there is? It's so great. It's a really, really good song. Love it. Enjoy, Justin. I hope you enjoy the song and blowing your nose. And I'll see you all next week. Yes? See you next week, Justin? You're listening to it right now.
Is that all there is?
Is that all there is? Yeah, it's so good. The end of the song is my favorite. Oh, Eric Knapp, you know it. Yes. It's really wonderful right now. It's really, really wonderful at this moment in time. Everyone, yeah, Justin, I knew you'd love it. Say goodnight, Justin.
All. Good night, Justin.
Good night, everyone. Good night, Kiki. Everyone, stay well, stay healthy, right? Stay safe, stay smart, and stay curious, and stay lucky. We'll see you again next week.
Keep dancing.